And the whole genome sequencing was derived from a spleen from the founders. We got a high coverage paired end reads, 150 base pairs from one sample per substrain. And the RNA-seq was derived from hippocampus, as I mentioned, the data is 50 base pair single end reads from 6 to 11 male samples per substrain. The whole genome sequencing data was analyzed by calling variants, SNPs and indels, structure variants with two different softwares, lumpy and c-aminators, and merged the structure variants. The gene dosage in or the segmental duplication dosage was inferred from depth signal in sec-dup annotations, and STR was also were also called by Hipster. RNA-seq data was normalized, and we found about 300 DE genes among these substrains. And following that, we did association studies with linear mixed models using genomic relatedness matrix to control for relatedness in these samples. And significant associations was inferred by FDR corrections. So the first observation, we looked at the distribution of different variants in these call sets, and we found variation hotspots present throughout the genome in certain segments. And all of these variations were clustered in the same or similar regions around the genome. You can see chromosome 4, 8, 11, and so on. So we asked, what is the significance of these regions? So if we segregate each variation type among different substrains, we saw that these variations actually come from B10 substrains, and all of them are common between these 5 B10 substrains, and they consist of 28 segments, which comprise only 5% of the genome and common in B10 substrains. And interestingly, about 90% of total SNPs was actually B10 specific. So the question was, where do these segments come from? So next, we looked at the SNP data from MGI and compared our B10 specific SNPs to them. And the data set from MGI has about 88 substrains from domesticus and musculus origin. So we required at least 300 common SNPs in each of these segments, and more than 90% genotype concordance. And here on the image in the y-axis, you see 24 segments past this requirement, and they have each segment have similarity to some substrain from MGI database, and we can see that longer segments, we saw fewer matches among the database. And this may infer that these regions are integressed from a newer so the integration that we hypothesize that these SNPs are from an integration process during the breeding stages, and the larger, the longer segments can come from a newer event, and the older, the smaller ones can, which are common between more substrains, they can come from an older event. So this was the hypothesis that these segments coming from some errors or some problems in the breeding stages and integressed into the genome of black 10. The next, we looked at association between expression data and variations that we found. So genomic variation was tested was loss of function, SNPs and indels, gene intersecting SVs, and gene intersecting seg-dupes and STRs. And as I mentioned, we use LMM, and the FTR correction was done with Benjamini-Hochberg method. And we found 53 significant genes, and they are, as you see in the QQ plot, they are very much enriched in seg-dupe regions. And a dozen of SVs and indels was also significant. And as you can see, we compared these associations with missense SNPs and indels, and they or intergenic SVs and STR, and they actually fall onto the straight line. So about the causality of these associations, generally we can claim causality. However, in some cases, we have a strong prior for causality. For example, in this gene, in KCC2, we found 6J-EIJ has a lower expression than all the other substrains. And among the significant cases or associations, there is one indel which is a frame shift loss of function. So this particular variation has a larger probability to be causal. This is one case of that we can infer some causality in these associations. Another mechanism for these associations is a gene dosage in seg-dupe regions, and as I mentioned, this signal is the strongest in our QQ plot, and most number of significant associations are these cases. And the mechanism is that in seg-dupe regions, the genes have different dosages in different substrains. And as you can see, the number of copy numbers associated with the expression in these substrains. Another interesting case that we found was a duplication in 3 exons in WDFY1 gene. So the middle 3 exons is duplicated in 6J and also present in the reference. And this is associated with expression reduction in 6J. And we asked if the additional exons introduced in the middle of the gene can it cause nonsense mediated decay. So to address this, we compared nonsense NMD impaired samples or embryonic stem cells expression data from wild type data. In the next slide. If we look at the sashimi plots for these samples, so the top one is BOMTAC, which doesn't have any junctions between exon 4B and 6A because it doesn't have the duplication. The black 6 has some junctions here. The wild type in the embryonic stem cell expression data has some junctions, but the knockout sample has a lot more junctions. And first of all, we can see that in the bottom right figure, we can see that the knockout expression for WDFY1 is significantly larger than the wild type. And also, if we look at the normalized number of junctions between these two exons, we see that the knockout has significantly larger number of junctions between these two. So we can infer nonsense mediated decay is likely the mechanism for decreasing the expression in the wild type sample in black 6J. So in conclusion, the different genomic variations were found in 14 closely related black 6 and 10 substrains. 28 potentially intergressed regions were found, introduced in black 10 substrains, and associations tests were performed in expression and genomic variations, and 53 gene associations were found with significant association, and a duplication of 6J and reference genome was attributed to nonsense-mediated decay mechanism. So the data is available. So the manuscript is available in bioRxiv. The whole genome sequencing FASTQ files is available in SRA, and process data you can access it in Mendeley database. Wanted to thank Dr. Palmer and Palmer Lab for helping to analyze and helping to process this project. Also, Jonathan Sabat had a significant role in this project. Also, Dr. Jim Breck for STR annotations and STR calling, and Dr. Wilkinson for providing the embryonic stem cell data to us. Thank you. I'll be happy to answer questions.